Good day to everyone. We are the Group 5, and today we are going to discuss the Chapter 11, Gene Therapy. This topic isn't just a chapter in a book, but it also represents a pivotal moment where technology and humanity cross to address the most fundamental aspects of our existence, our genes. So these are the objectives we are aiming to cover, and before we talk about gene therapy, we must first understand the concept of biotechnology. So, biotechnology, in its simplest form, is a technology that uses biological systems and living organisms to create products. It is a massive field of study with tons of applications, such as energy production, creating processed foods, waste management, developing genetically modified crops, and most relevant to our discussion, medicine, specifically therapeutics and diagnostics. So the next slide showcases a visual summary of the vast applications of biotechnology. You can see how it spans across agriculture, healthcare, the environment, and the industry. So in the healthcare, specifically, you can see treatments such as rDNA products, vaccines, diagnostics, monoclonal antibodies, and importantly, the use of stem cells and tissue-specific delivery methods, all elements that are crucial to advanced gene therapy. Now we talk the biotechnology. Now let's discuss about gene therapy. So gene therapy is a technology that allows us to manipulate the genome to correct defective genes and treat heritable diseases. Think of it as a precision surgery on a DNA level. It works through a process of gene editing, in which this precise process involves inserting good genes or correcting the faulty ones to prevent disease. The key takeaway is that it takes a specific DNA sequence. In fact, the potential scope of therapy is staggering. In fact, it treats major diseases shown in the slide, such as cancer, heart disease, and AIDS. Also, it directly addresses specific inherited conditions, such as hemophilia, cystic fibrosis, and severe combined immunodeficiency. While this is a breakthrough, remember that it remains a subject of controversy, which will be discussed later on. Now, for us to understand better and appreciate about gene therapy, we must first review the basic steps of our genetic material, starting with our genes and the human genome. So, let's talk about the DNA itself. So, DNA, or the oxyribonucleic acid, we can call it the blueprint of life because it carries the complete genetic instructions for every living organisms. Visually, it is that famous double helix shown in the picture, the two long strands twisted together. The key pieces of information are the four chemical bases in the center, which are the adenine, thymine, guanine, and cytosine. These bases form the rungs of the ladder, and their specific sequence is the code that gene therapy is designed to edit. Now, let's talk about genome. So, if a DNA is a single instruction manual, the genome itself is the entire library. It is the complete set of DNA that contains all the genes a body needs to function. When we talk about the human genome, we are talking about the DNA that are being organized into 46 chromosomes, which make up the 23 pairs, in which majority of them are non-sexual chromosomes, which we call them autosomes. Now, looking at the picture, this is a karyotype, a map of all our chromosomes. Notice how they come in pairs. This is because you inherit one chromosome from your father and one from your mother for each pair. This is critical because sometimes it only takes a one faulty copy from a parent to cause a disorder. That's why we need gene therapy. So the next slide showcases the structure of a chromosome, in which it consists of a long arm, a short arm, a centromere, and a strand, which we call them chromatids. So the next slide showcases of our DNA that is tightly wound are the protein, which we call them histones, in order to form a chromatin. And the next image shown on the right showcases the homologous pair of chromosomes you see here, which showcase the paternal and the maternal contributions. So when we talk about gene therapy, we are aiming to fix a defect or a single letter that is manually wrong, which causes a disease that may have been inherited from either of these original copies.
So, in continuation, I will be reporting the topic called genetic disorders. So, as we all know, each human chromosome contains genes or DNA sequence responsible for coding proteins. And these proteins carry out mostly the body's function, such as building tissues or defending us from diseases. So, basically, changes or mutations in this DNA sequence can cause genetic disorders. So, there are several factors or causes of why genetic disorder occur. First is the errors in DNA replication. So, mistakes during DNA replication can change the genetic code, leading to mutations that disrupt normal protein function. And the second one is environmental factors, for example, like diet, lifestyle, radiation, or toxins. And external agents can damage the DNA or affect how genes are expressed, which increases the risk of genetic abnormalities. So, the other two is chromosomal abnormalities or extra or missing chromosome, and the other one is mutations in a single gene. So these two will be elaborated more when we tackle about the classification of genetic disorder. So what are the effects of genetic disorder? So the effects are DNA changes alter the proteins coded by genes, and affected proteins lose normal function, leading to disease. That's why there is what we call gene therapy, which aims to correct faulty DNA sequences and help treat or prevent genetic disorders by restoring normal gene function. So, there are three classification of genetic disorder. First is single gene disorder, second one is chromosomal disorder, and the third one is multifactorial inheritance. So, in single gene disorder, it occurs when mutation or change happens in the DNA sequence of one specific gene. It is caused by a defect or mutation in a single gene that alters the function of the protein it codes for. So this disorder can be dominant or recessive depending on how the altered gene is inherited. So an example of a single gene disorder is what we call autosomal gene disorder. It's caused by mutations in genes found in the autosomes. So, autosomes are the chromosomes that control non-sex related traits and make up most of your genetic information. So, an example of traits is like your eye color, your hair color and its texture, and also your blood type. So, an example to this single gene disorder is a sickle cell anemia. A sickle cell anemia is a genetic disorder where some red blood cells are shaped like sickles or crescent moons, which can slow your blood flow and cause pain crisis, organ damage, and infections. The other examples are cystic fibrosis. In cystic fibrosis is it is a genetic condition that changes a protein in the body. The faulty protein affects the body cells, tissues, and the glands that make mucus and sweat. That's why, as you can observe from the picture of the affected of this genetic disorder, its airways are more narrower because of the formation of sticky mucuses. And the other example is what we call Tay-Sachs disease. In Tay-Sachs disease, it is caused by mutation of a single gene on chromosome 15. The defective gene stops the body from making enough of an enzyme. Without this enzyme, fatty substances build up in the brain and nerve cells. This build-up damages the nervous system, leading to muscle weakness, loss of movement, vision, and hearing loss, and eventually severe severe brain damage. So Tay Sachs usually appears in infants and sadly is often fatal by early childhood. So the other example of a single cell gene disorder is what we call X-linked disorder. It is caused by mutations in genes located on the X chromosome. These disorders are more common in males since they have only one chromosome. An example to this disorder is what we call hemophilia. Hemophilia is a rare disorder which the blood doesn't clot in the typical way because it doesn't have enough blood clotting proteins or clotting factors. So another example is what we call color blindness. Color blindness or color vision deficiency happens because special nerve cells in your retinas are missing or don't work properly. So this is reasonable because retinas are composed of various proteins, and in genetic disorder, DNA sequence are mutated which affects its production of protein. So the other example is Duchenne muscular dystrophy. In Duchenne muscular dystrophy, mutations in the dystrophin gene lead to progressive muscle fiber degeneration and weakness. That's why, as you can observe from the picture, the body composition of the affected is different from the normal body composition, which, as you can observe, an awkward arms and shoulders, curved back, weak buttocks, bent knees, big calves, protruding belly, thin weak thighs, poor balance, and falls often, walk on tiptoes. So I think that's all for my part, and the other two classifications and the other topic will be reported by the other member.
For the continuation of the classification of genetic disorders, we have here for number 2, chromosomal disorders. This happen when there's a change in the number or structure of chromosomes. So these chromosomes carry all our genetic information. Like they act as instruction manuals that tell our body how to grow and function. So we have here the karyotype of a normal male. Basically, the 23rd pair tells us the sex chromosome. So for male, the sex chromosome is XY, and for the female, as we can observe here, the 23rd pair is XX for normal female. But sometimes, this normal karyotype during cell division errors can occur, and this can result in having too many, too few, or rearrangement in chromosomes. So when that happens, it can disrupt normal development and lead to physical or mental abnormalities. We have here common chromosomal disorders. First, we have Klinefelter syndrome. This disorder affects males only. They have one extra X chromosome, and some features include tall height, small testes, infertility, reduced testosterone, lower IQ, and sometimes breast development. Although it's not inherited, it just happens randomly during the cell division. So next we have Turner syndrome, or also known as 45X or XO. This one affects females and they are missing one X chromosome. The common features are short height, a web neck, underdeveloped ovaries, leading to infertility and heart problems. Next is the most common chromosomal disorder. We have Down syndrome or trisomy 21. Down syndrome is caused by having an extra chromosome 21, and it affects both males and females, and the risk increases with maternal age. People with Down syndrome often have a short stature, flat facial features, heart defects, and delayed physical and speech development. Then we have Patau's syndrome or trisomy 13. This is caused by an extra chromosome 13. Some features include cleft lip, or palate, extra fingers or toes, small eyes, and severe brain defects. Unfortunately, most infants with this condition do not survive beyond the first few weeks or months. Then we have Edwards syndrome or trisomy 18. This is caused by an extra chromosome 18. Symptoms include small body, low set ears, clenched fists, and heart defects. And most infants with Edwards syndrome die within the first year due to many organ complications. So we are now down to the third classification of genetic disorder. We have multifactorial inheritance. Aside from chromosomal disorders and single gene disorder, not all genetic conditions are caused by a single gene or chromosome error, and some diseases are a result of many genes working together combined with environmental factors, and this is what we call multifactorial inheritance. Examples of multifactorial disorder include diabetes, which can be influenced by genes and diet, heart disease, and hypertension, affected by both family, both family history and lifestyle, asthma and obesity, linked to genes but also triggered by environment. Lastly, we have cleft lip or cleft palate caused by both genetic and maternal factors during pregnancy. Now let's move on to how gene therapy works. So, gene therapy, according to Bautista et al. 2018, is a method of treating or preventing diseases by replacing, repairing, or turning on or off specific genes in a patient's cell. It helps fix genetic disorders caused by mutations that affect normal protein function. So the main goals of gene therapy are replacing abnormal genes with normal ones, repairing defective genes, changing how a specific gene is regulated. And the most common approach is inserting a normal gene into the genome to replace a faulty one. So now, let's talk about the methods of gene therapy. There are two main methods of gene therapy. We have viral and non-viral. So for viral methods, this uses modified viruses that act as vectors to safely deliver the new gene into the cells. For example, retrovirus, adenovirus, and herpes simplex virus. For the non-viral methods, on the other hand, these do not use viruses. So instead, genes are introduced directly into the cells. For example, by injecting the naked DNA directly into your genes. So this method is much safer and easier to produce on a large scale compared to the viral method. And that is all for the classification of genetic disorders and how gene therapy works.
Following through, let's discuss the types of gene therapy. First is the somatic gene therapy. Somatic gene therapy is a medical treatment that introduces genetic material into a person's somatic cells or body cells to treat diseases. Somatic cells are non-reproductive cells like liver, lung, bone marrow, and muscle cells. The genetic changes made through this process are confined to the individual and are not passed on to offspring. The therapy can involve transferring a functional gene to replace a defective one, or using gene editing tools to repair our genes directly in targeted cells. The purpose of this gene therapy is to produce proteins or enzymes that the body cannot make due to the genetic defect. So somatic gene therapy is like repairing a broken part in a machine, and then the scientists replace the faulty gene in your cells so your body can work properly again. The repair stays only in you and not in your future children. Second is the germline gene therapy. are also called as a reproductive gene therapy. It is the modification of DNA in reproductive cells like sperm cells and egg cells, or in early embryos, what we call as zygotes, making genetic changes that are heritable and passed on to future generations. This process holds the potential to prevent and cure inherited diseases, but it is currently banned in many countries due to significant ethical concerns and safety risks. The purpose of this gene therapy is to correct defective genes in the germ cells before fertilization or early development, and to prevent the transmission of genetic diseases to offspring and future generations, and lastly, to eliminate inherited diseases permanently from a family line. So instead of treating the disease in one person, germline therapy prevents it from existing in future descendants. So, for example, if a genetic mutation causes cystic fibrosis, germline therapy could modify the embryo's DNAs that the child and all their future offspring will not inherit the mutation. So the key challenges of gene therapy is that the medical experts who did the procedure must correctly identify the genes responsible for the progression of the disease. Then the modified gene must reach the cell or target organ accurately controlled and effectively. Medical experts must also note that the cells can reject modified genes because of the body's natural defense mechanism that causes most of the gene therapy failures. So, next, let's discuss the different approaches of gene therapy. First is the ex vivo approach. It is a technique where cells are removed from a patient's body, genetically modified in the laboratory, and then reintroduced into the same person after the desired gene has been successfully inserted. The goal is for these modified cells to start producing the correct protein or enzyme that the patient's body lacks. So, one of the first successful examples of ex vivo therapy is the severe combined immunodeficiency. Doctors removed bone marrow stem cells from the patient, and a normal adenosine deaminase gene was inserted into these cells using a retrovirus vector. The corrected cells were returned to the patient, restoring immune function. Second approach would be in vivo approach. It is a method where therapeutic genes are delivered directly into patient's body to correct or replace a faulty gene, often using a vector like a modified virus. Unlike ex vivo therapy, where cells are modified outside the body and then reintroduced, in vivo gene therapy skips that step. The genetic material is introduced straight into the body using a vector. So, what are vectors? So, vectors are delivery vehicles used to transport therapeutic genes into a cell to treat a disease. They are typically derived from viruses, which are modified by removing disease causing genes to make them safe. So first type of vector is non-viral vectors. Since there are synthetic or natural delivery systems that are not derived from viruses. These methods are advantageous for being less likely to cause an immune response, having lower production costs, and the potential to carry larger genetic material, although they traditionally face challenges with efficiency compared to viral vectors. And lastly, viral vectors are modified viruses used as a delivery vehicle to carry therapeutic genetic material into a patient's cells to treat or prevent diseases.
Now, let's move on to the benefits and disadvantage of gene therapy. For its benefits, first, it offers a cure for various serious diseases like cancer, Parkinson's disease, AIDS, asthma, diabetes, heart diseases, and other hereditary disorders. This makes gene therapy a revolutionary tool in modern medicine. Second, gene therapy can replace defective cells, allowing normal cell function to be restored. Third, its potential extends beyond medicine. Gene therapy also shows promising applications in agriculture, such as developing disease resistant crops or improving animal genetics. Overall, these benefits highlight gene therapy's potential to improve health and quality of life. However, gene therapy also comes with disadvantages. One major concern is that Modifying an organism's genes can alter its natural capabilities, leading to unpredictable results. Another risk is that viral vectors, which are used to deliver the new genes, might regain their ability to cause diseases. There is also the danger of damaging the gene pool, which could result in multi-gene disorder if not carefully controlled. Furthermore, gene therapy is very expensive, making it inaccessible for many people. And finally, it raises ethical issues, which bring us to the next part. According to the US National Library of Medicine, several ethical questions raised from the use of gene therapy, which are: how can we distinguish good and bad uses of gene therapy? Who decides which traits are normal and which are considered disability or disorder? Will only the wealthy have access to gene therapy due to its high cost? Could the widespread use of gene therapy make society less accepting of people who are different? And lastly, should gene therapy be used to enhance human traits like intelligence, height, or athletic ability? These questions show that while gene therapy is scientifically powerful, it also requires careful and moral consideration. In conclusion, gene therapy holds tremendous promise for the future of medicine and human health. It has the potential to treat and even cure diseases that were once considered untreatable, offering hope to countless patients and families. However, this innovation can also bring serious challenges, including technical risk, high costs, and unresolved ethical questions. Scientists and policymakers must work together to ensure that gene therapy is developed and used fairly and responsibly. Society must also discuss where to draw the line between therapy and enhancement so that this technology benefits everyone and not just the privileged few. Despite the concern, gene therapy represents a remarkable step forward in biotechnology and could reshape the future of healthcare as we know it. Thank you for listening.